Welcome, AP Biology students, for Unit 6 about the DNA. We talked about in Unit 5 and before, like the nucleic acids, hereditary material. We talked about mitosis, meiosis as the hereditary material separates and go through crossing over in meiosis 1 in prophase. So we talked about so much about the DNA, and we talked about so much about the proteins, but how do they relate to each other? The DNA is the blueprint for the workhorses for the proteins. Proteins are important part of the cells and organisms because they are doing the work, transporting materials in and out from the cell, the carrying oxygen in the form of hemoglobin. The muscle cells contain actin and myosin as proteins. Antibodies are proteins. So we have so many different type of proteins that we have to really put together the picture how the DNA relates to the protein. So the DNA is the blueprint. And I just said the DNA in eukaryotic organisms and in prokaryotics too are carrying the hereditary material for the cell. This is the double helix. Later on in viruses, we will distinguish RNA viruses, single-stranded, double-stranded, single-stranded DNA, and double-stranded DNA, but it will come later. So the DNA is double helix, and it's made up of monomers, small repeated molecules, or the nucleotides. They are very similar to each other. They are different only in their nitrogenous base. So they are made up of the sugar molecule, the phosphate molecule, and the nitrogenous base. So I would like to draw a nitrogenous base for you guys to be able to see how they look like. This is the oxygen. This is going to be the sugar part. The sugar forms a closed segment. These are carbons. Carbon. And we have to number the carbons in here that we would know which carbon we are talking about. This will be carbon 1, carbon 2, 3, 4, and 5. And each sugar molecule contains the carbon 1 in the sugar molecule contains the nitrogenous base. I put an N in here because they contain nitrogen. In the DNA, this nitrogenous base be adenine, thymine, cytosine, and guanine. For the DNA, the carbon, second carbon contains only hydrogen. If it would have OH, that would be RNA, but no oxygen in here. That is why it's deoxy taking off an oxygen on here. The carbon, the third carbon contains a hydroxyl group. This will be super important for the reaction. And the fifth carbon contains a phosphate group. Okay, it's always like that, simple. Now we have another nucleotide, DNA nucleotide, that I would like to show you how they combine together. So we have another nucleotide, carbon, carbon, oxygen, carbon, carbon. Here's another carbon. I will add the nitrogenous base in here. And I will add the OH in here and the hydrogen. Oh, honestly, the third carbon is the important. And for the fifth carbon, I add one phosphate. Actually, I will add two more because this is how you would understand which part is combining with the other part. So if I add two more phosphates, they have huge energy in here. This is going to be a dehydration synthesis reaction as I combine these two monomers together to form a polymer anabolic reaction. It requires energy. The energy will come when we take off these two phosphate groups from the nucleotide, and they will, this phosphate group is going to form a dehydration reaction with the hydroxyl group. So water is going to leave the reaction. Did we learn that? Does it sound familiar? Yes, it probably does. It was unit 1, as far as I know, when we learned about the macromolecules at the beginning of the biology journey. So that's why it's super important that the new nucleotide can only be added to the third carbon. The third carbon cannot add to the fifth end of the nucleic acid. So just only this way. Okay, the phosphate always combines, the phosphate groups always combine with the hydroxyl, but not vice versa. Okay, so we did this. I tried to check what else we need to talk about. So the base pairing. So for the DNA, if we have, we distinguish two strands, the coding strand and the template strand. Coding strand. So I come up with an example for that: A, T, C, T, G, A. And I will say that this is the third end, and this is the fifth end. I'm referring to the third end and the fifth end, how they orient with each other. I can figure out the template, the other strand of the DNA. Adenine combine always with thymine, and they form, I will just put it in here because you need to know it, two hydrogen bonds with each other. Thymine combines with adenine. Again, two hydrogen bonds connecting those nucleotide nitrogenous bases together in the nucleotides. C combines with the G, cytosine with guanine, thymine with adenine, guanine with cytosine, adenine with thymine, and this will be the third. And I would like to point out that the thymine and guanine, they form three hydrogen bonds with each other. The adenine and thymine only two, the cytosines and guanine, and here too, they form three hydrogen bonds with each other. Now, I just used dotted lines because they are not solid like that. These are covalent bonds, but in here, they are hydrogen bonds. It will be, so an enzyme will be able to unzip the double helix when it requires replication. Okay, up till then. So, classic example for that. If a DNA strand contains 20% thymine, the question is, what percent will be the adenine, guanine, and cytosine in that DNA segment? We know that in the DNA, thymine is 20, so it has to be 20. The adenine, altogether, it's 40%. 100 minus 40 is 60%. So the 60% is between the guanine and the cytosine divided by 2. So we can say we have 30% guanine and cytosine in that DNA segment. Now, let's get into the replication. And I would like to draw out a strand. So here is the DNA that needs to be replicated. So this is the third end. Oh, we did not just set in here. So please notice that the third and fifth end for the coding strand, and for the template strand, for the opposite, for the complementary one, it's going to be opposite. So they are anti-parallel strands. So wherever the three ends, the other strand has its 5 and the phosphate group facing opposite to the hydroxyl group. This will come really important to figure out the leading and the lagging strands. So this is the third end, and this will be the fifth end. We will just name it when they are separated from each other. They will be the fifth end, this will be, and this will be the third end. Let's say we have adenine, thymine, cytosine, guanine. These are the nucleotides. C, G, C, G, T, okay, and in here, now I have to just be careful and just matching the pairs up. T, A, T, A, C, G, C, G, A, T. Now, something really opened this up, and we have one enzyme first, the topoisomerase, that is going to keep this replication fork, that when we have an enzyme in here, this will be the helicase. How do I know that this is enzyme? Because it ends with an ase. All of the enzymes except the one of them in the plants is ended by ase. That's why you know it. Helicase is opening up the strand. Now, we have, I will simplify pretty much how is going to be like that. So this end is, we need a T in here. The T has a so, okay, we will start from fifth end. Wait for a sec. So, no, we have a T in here and contains a phosphate end in here and an OH end here. A new nucleotide that is all around in here, all around what we eat comes with that, and your body can make it, OH, and the G with a phosphate group Well, these are all three of them actually. There are three phosphate groups to provide energy for the dehydration synthesis. So here is going to, we need an adenine. This will come here and combines with the thymine in here, so it adds up in here. We will have another thymine in here with the phosphate end. This is the fifth end, is going to just combine in here, and an A and a C and a G and a C and a G. So it will grow this way. As the helicase opens up this replication fork, the replication of the nucleotides on that strand are going to follow the helicase. It goes this direction. That's why we call this strand as a leading strand. The other one, on the other hand, is a little bit different. This is going to start here, for example, like a G in here, because the G has the OH in here, and the phosphate is here. Thinking about this. Okay. No. This is where the phosphate is, and this is where the OH will be. Okay, that's a different one. So this is a G. And now, again, we can add only to the hydroxyl end. This is the third end. So we will add a C in here when it contains the phosphate group in here and OH group in here. We add, again, a G in here where the hydroxyl group is here, and we can add only to this end, but not the phosphate end. So it goes the opposite way as the leading strand. It goes away from the helicase, from the replication fork. Now, this is going to be the lagging strand because it's moving away. So when this helicase will open it up more, another enzyme has to come and start again. This is facilitated by the RNA polymerase 3. No, DNA polymerase 3. So the DNA is going to just be pulled in the nucleotides, and the DNA polymerase are combining that, facilitates that dehydration synthesis. Now, this is not that simple as it is now, because we will have a little bit more complication. So we talked about the topoisomerase, that keeps everything open. We talked about the helicase that separates the hydrogen bonds between the nucleotides. What else we have? The DNA polymerase 3 is making the lagging and leading strands. That facilitates the dehydration synthesis. We also have another stuff like the DNA polymerase cannot start putting the nucleotides, the complementary nucleotides in a row. It needs a primer. The primer is made up of RNAs. So in here, we did not talk about the RNA. The RNA is very similar to DNA. Carbon, oxygen, carbon, carbon, and carbon. And we have a phosphate group in here, a nitrogenous base, and OH in here, and OH group in here in the third. So now the nitrogenous bases can be adenine, uracil, cytosine, and guanine. They do not have a thymine in here. That's really the difference. Besides, also the second carbon contains a hydroxyl group. So we have the RNA primase that will make a primer starting the lagging strand, starting the synthesis. And this RNA primer, first a primer, for example, in here, C, U, and A. Okay, this is the primer for that. And this primer will give a starting point for the DNA polymerase 3 to latch on and start to continue the synthesis. Now, this RNA primer, though, has to be changed, has to be taken off by the DNA polymerase 1. We'll switch the RNA sections in here on the DNA. And also in the lagging strands, because they will have segments, they are going to be called the Okazaki fragments. Those Okazaki fragments has to be binded together, and this is another enzyme, the ligase. The ligase is only in the lagging strand, binding together the Okazaki fragments. On the lagging strand. The lagging strand, too much going on that. I totally understand. So we are good, and the primer is changed by DNA polymerase 1. The whole easy job. So this is by DNA polymerase 3. The replication, it's one more thing about the replication is that it's on semi-conservative way. We have the original DNA, these are the originals, and they will separate by helicase, and new strands are going to be made in here. So the new cell contains one original DNA and one newly made DNA in the cell. That means semi-conservative replicating method. And one more thing we have to talk about, the organisms. Eukaryotic, prokaryotic organisms, they contain DNA double helix for their hereditary material. For the viruses, it's a little bit different. They can be double-stranded DNA. That's easy, kind of similar. But sometimes they have single-stranded DNA, or they can have single-stranded RNA and double-stranded RNAs. There are some viruses that they contain double-stranded. Now, the double-stranded DNA is actually the RNA virus. First, when the RNA virus is getting into the body, it needs to be translated in itself into DNA to integrate into the host DNA. And the enzyme that is making the RNA to DNA, please recall it, reverse transcriptase. That is a special enzyme in the viruses that is doing the conversion of RNA to DNA. Okay, that's all for now.